quantum computing will revolutionize pharma and chemistry as we are doing it today. This is what I heard at Y Combinator Startup School 2016 when I was listening to Chad Rigetti speak about quantum computing for the first time. And then again at their hackathon in 2018. And then again at the IBM Quantum Summer. And I'm sure many of you have heard this many times today. Today I'm going to walk you through some actual applications of quantum algorithms that can be used in pharmaceutical industry today. My name is Samarth Sandeep, and along with my co-founder Kirk McGregor, we are the founders of If and Only If Technologies. Our founding story begins in early 2020 when we developed an algorithm based on the QAOA max-cut algorithm for finding the most electronegative atoms in proteins, pruning the rest of the protein structure off, and using these smaller structures to increase the throughput of docking studies. So we decided to apply this algorithm in a COVID context, looking at how the SARS-CoV-2 ACE2 complex binds to hydroxychloroquine and azithromycin. And this study, to our surprise, was cited by Pfizer, the National Research Council of Italy, and many other top research institutions. So this led us to think, well, there must be a market opportunity here. What are the key problems the industry is facing today that maybe we can start to tackle? So after speaking with some of the top 10 pharma companies and with some top research institutes, we realized the problem was not lead generation, as many companies state, but really inefficient validation. So this is specifically a problem when you deal with diseases that work around membrane proteins. Membrane proteins are the ones that are found on the outside of the cells, and imaging them can take a long time, and statistical solutions such as AlphaFold can only get so far in terms of getting the correct complex because there is no data for it to take from the past. So our solution to this problem was a dynamics-oriented biochemistry platform. So we not only have our QAOA solution, we've developed our own literature analysis solution, our own protein binding software that's based off of Gaussian boson sampling, and then our own graph isomorphism search software that can help look for chemical homomorphisms. So we'll walk through some use cases now of how our software can be used. So one problem that was brought to us by a doctor at Mount Sinai Medical System and at the University of Kentucky was how to look at clinical trial picking from a biochemistry standpoint. So how to do it not just based on what disease that trial is looking at, but actually what are the proteins that are involved in it. So we used our binding algorithm to look at what the binding concentrations are and help rank these proteins to look at what are the most relevant trials. Second, we used our QAOA max-cut algorithm to figure out what are the most important atoms in surfactant proteins A and D, and then complete docking at high scale. So in this study, we compared our work with QAOA max-cut to the GROMACS temperature replica exchange molecular dynamics system for finding binding sites prior to docking. And we found that our systems got the same results, but that ours took 8 minutes and GROMACS took 6 hours, even when running on the Pittsburgh supercomputer and on the Jülich supercomputer. So another use case I want to highlight is we have a software that's based off of the graph-subgraph isomorphism problem that's being run on quantum annealers, where we can find chemical homologies. So specifically, we were brought to a problem from the Petroleum Environmental Research Forum, where they were trying to see how they could do oil cleanup. So we are able to both take their existing molecules that are found in oils and the existing solvents they use and find homologies that have the same chemical structure, but they have different structural alignments. So on the left in blue are the starting chemicals, and on the right in red are the homomorphisms that our system was able to come up with. So in terms of how we want to provide these services to clients, we're a pretty standard software company, but also we are looking to license our tools, such as a hypercomplex, native way to deal with hypercomplex numbers in Python with TensorFlow. That may be helpful for those of you doing ZW calculus. And then also our own HPC-ready containers that have our algorithms, as well as molecular dynamics solutions that currently exist, so you can do benchmarking. In terms of where we see this market growing, I mean, it is a very big market, pharmaceuticals, but more importantly, I want to stress that biosimilars as a market specifically is growing. It's the fastest growing sector in the pharmaceutical industry, valued at $452 billion just this year by the Journal of American Medical Association. So it's definitely going to be even more of a market in the future, considering that mRNA vaccines and genetic-based drug development is taking off. So having methods that can de-risk these new drugs that's not just based on protein and small molecule interaction, but protein-protein interactions at scale is very, very important. Thank you so much for listening to my presentation. At this time, we welcome any questions. Please reach out to my email or send me a text if you have any further comments. Thanks.
So you've stated you've done a lot of things with a lot of different people so far. What are revenues today?
So yeah, we do generate revenue. Not zero.
When I hear QAOA and you said that you got a speed up, I find that very unbelievable because QAOA has not proven to give a quantum speed up, especially at the number of qubits we have. I mean, this sounds very unbelievable. Do you have any scientific backing, and are there like well-known scientists who are going to back that you guys have a quantum advantage, especially the same quality of solution you can get from a classical computer?
So this study is actually was published in Frontiers in Immunology just a couple months ago. I didn't say that we were able to get a speed up over classical QAOA. I was saying QAOA as a method was faster than GROMACS TREMD for finding electronegative atoms. That's all I've been trying to say.
So not on a quantum computer?
So what we found was still that, yeah, doing it virtually was still faster than doing it on a quantum computer, which is in line with what everyone is finding.